Hello everyone and welcome to today's webinar, head-to-head comparisons using real-world data, design and data source considerations from pilot investigations in cardiovascular disease. My name is Alund Gupta. I am a data scientist in the real-world and advanced analytics group at Cytel, and I am leading this pilot project on cardiovascular disease. We also have with us Miguel Hernan from Harvard. He is professor of biostatistics and epidemiology at Harvard University, and he has laid down a lot of the foundation for this particular pilot project that we'll be speaking about today. Miguel is a well-known researcher in the field of causal inference, and he has published a great book that's available online. I think it's still free, so go check that out. And finally, we have Judd Schneider, who is CTO at Nashville Biosciences. Cytel is partnering with Nashville Biosciences, or conversely, they're partnering with us with their real-world data and expertise surrounding that data. And we'll hear more from Judd today on Nashville Biosciences more broadly, but also on sort of considerations for data, for real-world data in the context of trial emulation, but also in general, I think. I just want to remind everyone that this is a part of an ongoing series of webinars that Cytel is hosting on the two pilot investigations in target trial emulation. We had the first webinar July 7th, which is sort of an introductory webinar, and this one is number 2, which is on, as I said, design considerations and data considerations in the context of the CVD pilot. We'll talk about that at length today. And the next one will be sometime in August, which will be hosted by my colleague Devin Boyne, who is working on the other pilot project in pancreatic cancer. He's a little bit ahead of us, so you'll hear more about methods and hopefully even some preliminary results by next time. The date for that is to be decided, but it'll happen in August and we'll keep you informed. These webinars will be hosted in the summer and early fall as these two pilot projects progress in real time. So keep an eye out for those. So with the housekeeping done, I'll begin with an introduction on target trial emulation. This will be a perhaps not so brief refresher for people who attended webinar 1, but I just want to be thorough with this. And essentially what we're talking about is trying to emulate randomized clinical trials, which are the standard, in some sense, the gold standard for comparing effectiveness and safety of interventions. The randomized design by itself reduces selection bias and confounding. If you do things sensibly, you can reduce or minimize other kinds of biases that are likely to arise in other kinds of designs. And the outcome is that randomized trials provide causal estimates of effects, which is not, for example, what you'd get if you were doing some kind of predictive modeling, where at best you would get something that is hypothesis generated. Health policy and decision making rely and need to be informed by causal knowledge about comparative effectiveness and safety of different interventions. So the kinds of questions that you may ask, or one asks in general, is should we treat with drug A or B, or A, B, or C, or any set of possible set of drugs, or some more complicated questions about more complex treatment strategies or regimens, like do we treat with drug B or C if there's no response to drug A, or should we treat now or should we treat later? And also which individuals to treat, which individuals are most likely to benefit or show a response to a particular intervention, and which individuals are most likely to develop adverse events and should not be treated. But randomized trials are not always feasible, and they're often not available for all the kinds of questions that one might ask. Due to possible reasons related to cost, they're obviously costly. They can be untimely, so if you need to answer a question right now, you might not be able to wait for 5 or 6 years for a clinical trial to be completed to be able to answer that question. They can also be unethical, so if there's somewhat of a consensus that a particular intervention is effective, that it might be unethical to withhold that. And they can also be impractical. And often, even though you might be able to actually execute a clinical trial, they may be limited in scope in different ways. So you might have a more restricted eligibility criteria, again, due to some of the reasons that I cited, due to cost and time. They might have a smaller set of outcomes that are measured, fewer or not all the various possible interventions will be compared due to time and cost limits and so on. And there are also often questions about real-world generalizability or transportability of the effects that you estimate from a randomized trial. So if, for example, due to the eligibility criteria, if the population that's included in a clinical trial does not represent the wider population, then there are questions about whether the effects that you estimate from a randomized trial are likely to be generalizable to the wider population. And even if you don't have a clinical trial, decisions still need to be made, even in the absence of one. And maintaining the status quo is also a decision that you're making. So ideally, you need something to answer these causal questions in the absence of a randomized trial, or to be able to complement existing trials. So plan B is that we can try and emulate a randomized trial using real-world data or observational data. So just in the diagram at the bottom, you start out with a causal question. You ask whether a randomized trial is feasible to be able to answer this causal question. And ideally, this would be, and you go ahead and run the randomized clinical trial to answer your causal question. But in cases that it's not feasible, you try and emulate a randomized trial using real-world data, with the limitations that you cannot emulate placebo or blinding or forced adherence in these kind of sort of retrospective analyses. And you're also limited by the data that's available to you. So you can only work with or look at therapies that have been approved for use and are actually represented in the real world data that they're using. So the way we try to answer causal questions from real world data is using the Targetfile Framework that Miguel, amongst others, developed. And it occurs in 2 steps. So you start by asking a causal question, which is equivalent to specifying the protocol of the randomized trial that can answer the causal question of interest. And step 2 is actually answering that causal question by identifying a suitable real-world data set, source for that data set, and then trying to emulate the randomized trial using real-world data. So just as an analogy for these 2 steps, you start by setting a target. In step 1, setting the correct target, and then in step 2, you try to aim for the center of the target. And this is not a one-way street, so usually it is necessary to cycle and iterate between steps 1 and 2. For example, to tune your protocol that you set up in step 1 due to practical limitations. For example, because of the data that you have, for example, by restricting eligibility criteria to only include patients that may have more complete data at the end. And we'll talk a little bit more about this. Now, why do we need a Targetfile framework? Why can't we just analyze real-world data normally? I have 2 reasons here. The first is that you may end up asking the wrong questions, not the causal question that you're interested in. Or you may ask a poorly framed or poorly defined causal question. And that means everything that you do downstream is kind of wrong, or not actually answering the causal question that you're interested in. Whereas with the Targetfile framework, the explicit representation of the causal question as a target file prevents this from happening, or at least minimizes the risk if you set it up sensibly. And so we'll talk about today how to actually set up this protocol. Who needs to be involved and what you need to do to be able to frame this question correctly as a protocol. And secondly, you may have set up the causal question correctly, but you may be answering it incorrectly. For example, by not accounting for the various biases that may arise, that are known from research in epidemiology, exist in have existed in observational analyses. Things like selection bias and immortal time bias, which we talked about last time as well. or not adjusting for the relevant confounders, or adjusting where we shouldn't, adjusting for variables where we would introduce bias and adjusting for them, and also not using the appropriate methods. And so the effect of that is that you get biased effect estimates and you come to the wrong conclusions. And an example of this, so this is a rather old example, but I think it's really pertinent, is this question of looking at the effect of postmenopausal hormone therapy on the risk for developing coronary heart disease in women. So to answer this question, there was a randomized trial that looked at initiators of hormone therapy versus non-initiators of hormone therapy, the usual thing that you do in a clinical trial, that found that the hazard ratio was 1.24. So essentially, women who used hormone therapy, postmenopausal hormone therapy, had an increased risk for coronary heart disease. What I have here are two, or examples of two analyses using the same real-world, at least the same real-world database. The first I'm calling a naive analysis, which perhaps set up the question incorrectly. It was looking at users, prevalent users versus non-users, instead of the question you ask in a randomized trial, initiators versus non-initiators. And it found that the hazard ratio was 0.68, basically the opposite effect than the randomized trial. On the other hand, we have this work from Miguel from 2008, that's published in 2008, that used the Targetfile Emulation Framework, looking at initiators versus non-initiators, and they found that the hazard ratio was much closer to the one that was identified in the randomized trial at 1.05. So the conclusion is from this slide that you want to be able to work under the Targetfile Framework to be able to estimate the effects correctly by first setting up the question correctly and then answering that using the appropriate methods. Miguel, do you have anything else you'd like to add to this?
Only one general comment, which is that all this Targetfile Framework, which, by the way, has existed for decades, even if not with this name, but people have been thinking in terms of how to use observational data to emulate a randomized trial, and there is a long history of that since the 1950s, at the very least. Although we gave it this label, but we didn't come with the idea. I don't want to take credit for that. But the main point is that if you think about a conventional analysis of observational data, you will see something like, well, you know, for each person you divided up the data into person years, and now each person year is either an exposed person year or an unexposed person year, and now you compare the incidence rate in the exposed person time with the incidence rate in the unexposed person time. That, which is how many of us have been taught epidemiology, it's a disaster. That's a recipe for bias, because once we start doing everything in terms of exposed person time, unexposed person time, that classical way of analysis of observational data is doomed, and that is exactly what happened in the example that Alan was describing here of the hormone therapy. The whole problem is that once we start thinking in terms of person time only, then we lose the relation to the causal question, because the causal question is not about person time. The causal question is about what happened if in this group of people, of persons, not of person time, of persons, if they were taking this treatment from this time 0 until the end of follow-up versus taking this other treatment from this time 0 to the end of follow-up. All that is structure that is essential in any clinical research project, like in a randomized trial, is completely lost once we start talking about exposed person time versus unexposed person time. So the Targetfile Framework brings back the key elements of a study design that we all accept as valid in randomized trials, bring those elements back into observational data. Thank you.
So just for some use cases for target trial emulation and the kinds of things that in the real world such an approach could be used for, You can use it for generation of efficacy or safety evidence, for conditional regulatory approval, or post-market assessment, providing a comparison when network meta-analysis is not possible. So for example, when you have disconnected networks, expanding the scope. So for example, expanding the eligibility criteria of a randomized trial, refining aspects of an existing treatment protocol to optimize it perhaps, and enabling comparison to identify optimal treatment regimens. Okay, so for the rest of the talk, we'll be focusing on the pilot project on type 2 diabetes, or head-to-head comparison of second-line therapies for type 2 diabetes. And we'll be discussing explicitly really the considerations for protocol design, for how you design the protocol to address a causal question. How is it different from designing a randomized trial, perhaps? And also, what is a suitable data source? We had some questions about this last time as well. So just as sort of a background for this project, so the unmet need in this area, patients with type 2 diabetes usually start out on metformin. It's got a good history of use. It's got a favorable safety profile in most cases. It's effective. So metformin is usually given as the first line of therapy for these patients, but a lot of patients often fail to achieve or maintain glycemic control, or sufficient glycemic control, whatever that might be, and require second line therapy. And currently, there are more than 6, actually around 10 classes of anti-diabetic drugs that have been approved for use without a head-to-head comparison of effectiveness and safety. And the choice of second-line agent, what second-line therapy is given to patients, varies across clinical practices across the world. So there's a need for comparative effectiveness evidence to guide the choice of second-line therapy, to reduce detrimental effects, to improve cost-effectiveness, and to guide personalized therapy. This is just results from the Global Discover study that looked at prescription practices in diabetes in general, but this figure shows that efficacy is the main driver in prescription practices globally. So basically what this graph shows is the reason cited by investigators for prescribing a particular second-line therapy for people with type 2 diabetes. And efficacy is the most important. And so it's important for us to be able to address this issue. So we start by asking the causal question. This is step 1. And as I said, this is equivalent to specifying the protocol of a randomized trial that can answer this causal question. And as I said in the beginning, the groundwork for this particular project has been laid in this white paper from PCORI, Patient-Centered Outcomes Research Institute, which was developed by Miguel in collaboration with others. So basically, I want to bring in Miguel at this point to talk about how do you actually go in practice to actually get the protocol design for if you want to do a target trial emulation study. So the considerations, and Miguel, please speak to these points. Who needs to be involved in setting up this protocol? What are the roles for those involved? How do you define the data and the variables that are necessary for emulation? And at the end, how do you refine the protocol given practical limitations after you've identified the data and so on?
Thank you, Andy. So the key point is that we need to be able to design a randomized trial if we want to use observational data for causal inference. So even though we are going to be working with real-world data or observational data or any name that you want to give it, the first step is the specification of the protocol of the randomized trial that we are trying to emulate with the observational data. So the people who need to be involved in this step are exactly the same people that are involved in the design of any randomized trial. We need subject matter experts. This is not something that can be done by methodologists, by statisticians and epidemiologists left on their own. If we are asking a clinical question, we will need experts in the clinical question. So it's really not very different from any other aspect of clinical research. If we are going to ask a causal question, we need to know what is a randomized trial that would answer that question and specify the components of its protocol. What is different now is that because we are using observational data, that means that we're going to have some constraints of the data that are available to us, which implies that the design of the target trial is constrained by the data that we actually have. We cannot say, oh, we are going to emulate a target trial in which we have people between the ages of 40 and 80, if in our data we only have people over 65. So our target trial is going to have to be among people over the age of 65, even though we might have wanted to look at younger people. So the target trial is not the ideal trial that we would like to conduct. The target trial is the trial that we can argue with a straight face, that we can emulate with the data that we have. And if that target trial is not what we want, we think that that's not going to answer the question that we want to answer, because we don't have enough data, because we want to answer the question in people age 40 and we have data of people over 65, then we have to quit and look for a different database. So that is really the different part. It's not the design of the target trial, which is the same as any design of a randomized trial. Is the part that we have to live with those constraints that exist in the real world data, and make a decision whether those constraints are too much and we are not willing to accept them, or whether we are willing to accept them and design our trial there. But if we have the data on the eligibility criteria of interest, on the treatment of interest, on the outcomes of interest, then we are half the way there. The other half is having data on the confounders. And that's the data that we don't need in an actual randomized trial, because treatment is randomly assigned. Typically, we don't have to adjust for confounding. But when we're emulating the target trial, we need to emulate the randomized assignment. And that is what adjustment for confounding means. We adjust for confounding because we're trying to emulate randomization. If we could adjust for all the confounders, there will be no difference between an observational study and a randomized trial. So that means, again, that we need to involve in the design of the analysis of the observational data people who know people who know which the confounders are. And that means, if we're asking a question like in this particular example is about the effects of treatments for diabetes, then we will need to involve clinical people who know why some people get one treatment versus the other. So if we have data on eligibility criteria, treatments, outcomes, and we have data on the confounders or on the main confounders, then we will be able to emulate the randomized trial, or approximately emulate the target trial. Thank you, Eddie.
Thanks. There was a question about something about a causal effect on outcomes. So we are asking what the causal effect of a particular set of interventions, for example, is on a set of outcomes. Yes. Thanks, Peter. So we'll go to the next one. So for this, actually, there is an ongoing randomized, actually a pragmatic trial based in the United States to address this same question that we're asking with this pilot investigation, looking at comparative effectiveness of second-line therapies in type 2 diabetes. It's called the GREAT trial, which started in 2013, and the estimated primary completion is next year, July 2021. And essentially, as you can see on this diagram on the right from the paper, so describing the trial. So they're looking at patients with type 2 diabetes who are taking metformin, who do not achieve their glycemic control targets as measured by glycated hemoglobin or HbA1c, and they're randomized into 4 groups. So there are 4 different possible second-line therapies that can take support: DPP4, GLP-1, and insulin, basal insulin. And they're looking at outcomes including primarily HbA1c, which also is the most commonly measured outcome for type 2 diabetes, at least in trials, but also on things like cardiovascular side effects, microvascular side effects, adherence, tolerability, and so on. Now, one of the distinctions for this study, and something that I think our trial emulation can sort of add to this trial, the GREAT trial, is that the GREAT trial was unable to include an SGLT2 inhibitor arm because they had just, I think they were not actually approved in 2013 when they started with planning the GREAT trial. And we will be able to include an SGLT2 inhibitor arm. It's an important class of second-line therapies, or actually anti-diabetics, that has been approved, that was approved, I think, 2014. So that's something that we can add to the trial, obviously. But essentially, what the GREAT trial provides us is a framework that we can compare our results against. We can benchmark our results against by using, hopefully, the exact same sort of eligibility criteria and the kinds of treatment strategies that we're looking at, and compare our effect estimates to theirs. And then we can also perform a parallel set of analyses with an expanded eligibility criteria, including things like SGLT2 inhibitor and so on. Okay. So we come to data next. So once, let's say, we have the protocol set up in the way that Miguel described, we need to look at data. What would be a suitable real-world data set that we need to be able to successfully try to successfully emulate a target trial, a randomized trial. So what I have here is just a list of some recommendations for real-world data. So ideally, data should be from a combination of medical records, electronic medical records, EHRs, claims data, clinician notes, etc., to be able to get a fuller picture of, for example, the patient history, because each of these are measured at a different time. They provide a different look at the treatment history and so on for the patient. Also, we want to be able to get big data. And what I mean by that is that we want sufficiently large sample size. So ideally for the GREAT trial, there are around 1,000, 1,250 individuals in each treatment group. Ideally, we want to be able to have a larger sample size in our trial emulation compared to that, to be able to estimate the effects more reliably, hopefully. We also want a large number of variables. So as Miguel talked about, we need to be able to identify confounders. We'll talk about what confounders for this project, so you can get a sort of an example of the kinds of things you would need. But essentially, we need large N, and we need large number of variables. We also want this real-world data set to have a representative sample that's covered from the population. So if you're getting it from, if you're using a single data set from, let's say, a hospital center, you want to make sure, hopefully, to be able to get an idea of how representative that is of the larger population that you're looking to represent in some way. You also want to look at bias or irregularities in the capture of study variables, especially as clinical practice, for example, changes over time. You want to have a data set that tackles that in some way. Obviously, you want a long follow-up for patients and a large number of, as I said, follow-up variables as well. And you also want database expertise. You want you don't just want the data, but you want people who know the data really well, who can identify where certain variables are, how to derive certain variables, and they have deep experience with using that database. Ideally, you also want access to clinical practitioners who are familiar with the data and how it came about. And there are also things related to data normalization, harmonization of different codes and so on for diagnosis and treatments, and also de-identification if you're looking to, obviously, if you're using patient data. And for actually getting your cohort at the end after you apply, or you filter this big data set through your eligibility criteria, you want to make sure that you address things like bias. So if you're selecting, for example, in this project, we're selecting for people with type 2 diabetes diagnosed in the past, you don't want to select patients who may have had a possible type 2 diabetes or had a test for testing for type 2 diabetes. So you want to be able to account for those. Things like false positives and negatives. So at this point, as I said, and I mentioned at the beginning, Nashville Biosciences is who we're partnering with for providing the RealWorld data set. And I'll bring in Judd at this point from Nashville Biosciences to talk more about Nashville Biosciences broadly, and also perhaps hit on some of the points that I talked about in the last slide.
Yes, thank you very much. So my name is Judd Schneider. I'm the Chief Technology Officer of a company called Nashville Biosciences. A little bit of history about us. We are actually a subsidiary of Vanderbilt University Medical Center based in Nashville, Tennessee. We are basically a subsidiary company that works with commercial partners who are looking to leverage a lot of the data and genetic assets that have been developed over time within the medical center. Primarily the one that we're talking about here is an asset called BioView. BioView is a database that it's a program that's been started about 15 years ago in the medical center as a way to capture all of the medical and clinical history of patients that went through Vanderbilt University Medical Center. It has been used in over 180 different peer-reviewed journals as a data set. So it's well published, well validated, and it's been used internally at Vanderbilt for the past 15 years, really, to power a number of different types of clinical research studies. One of the things that enables us to basically have such a large effect is that it really is an all-comers database. So if you come into the University Medical Center, you're first consented into the process, and then all the data is de-identified. And this is all different types of real-world clinical data. So anything from your first encounter with your primary care provider all the way up through your screening colonoscopy, or if you happen to interact with a specialist, all of that information is then captured and de-identified into that database. It's a pretty large database. It's about 3 million de-identified subjects and their entire longitudinal clinical history. We have an average length of about 12 years of clinical history per subject within the database. So it is pretty broad and pretty deep. Now, one thing to note about how we work with customers is that, as said prior, it's important to understand and how to work with the data. So a couple of things that are unique to our data set is, one, it's normalized into a format called OMOP, which basically allows us to standardize a lot of the specific clinical parameters into standardized concepts, and it allows for easier analysis. A second thing is that we actually work side by side with our customers to help them understand and use the data effectively. So it's not simply that we provide the data, but we actually provide that expertise that was mentioned earlier, both on the data side, but also we can reach into the medical center and consult with the clinicians who are actually treating patients day to day within the medical center, and provide key insights into how things work within Vanderbilt as opposed to another medical center, or how different clinical practices operate within the medical center. We also do, it's not relevant to this project, but we do a lot of bioinformatics. We have a large genetic component to the database where we have about 265,000 germline DNA samples, and we've got about 120,000 that are currently genotyped on a 2 million SNP array. We also have, what's unique about our database, we also have some image data too, which is relevant for different types of studies. Next slide, please. Just to kind of set the stage, though, as every medical center is unique within both geographic as well as its patient population. Within this particular study, we're looking at diabetes, and Nashville is positioned within the southeast U.S., right in the middle of Tennessee. But Vanderbilt itself is a large tertiary medical center within the southeast, so it's not that we're simply getting patients from the surrounding counties in Tennessee, but we actually have a broad reach within the surrounding states. And that's important because we're a center of excellence in a number of different areas. And we draw a lot of patients from these surrounding states who need to consult our experts for their health. Within that, we have about a reach of about 6,100 providers, and we touch about 68 hospital locations within the expanded Vanderbilt Health Network. Most of the data comes within the BioView database, and the database that we work with comes primarily from within the medical center campus that's based in Nashville, Tennessee. But we do have a lot of these other hospitals do refer subjects into or patients into our main hospital for a variety of different reasons. We touch about 250 million lives annually within the Vanderbilt Health Network, and all of that data then flows into our database, and that's a wide variety of different metrics. So that can be anything from your basic characterizations and ethnicity, gender, date of birth, but all the way through your clinical biopsy reports, your imaging reports, routine lab data, all of your CBCs, all of your different types of esoteric labs are in the database. We have medication use history, and that's fairly extensive. It's not just prescribed, but it's also dispensed within the medical center as well as administered within the medical center and patient reported, which can be useful in a variety of different types of contexts. We do have some information about insurance. We have information on your standard types of billing and claims information. We also have clinical notes that we routinely use to go back to the source to really understand some of those nuanced questions that was alluded to earlier of whether or not this subject just has this disease, or what disease process was started, or when it was started, or what was the thinking around a particular treatment at a particular period of time. For this particular study, we have about 165,000 different subjects within the database that have diabetes. The Southeast U.S. has quite a bit of diabetes, and Vanderbilt is one of the larger medical centers treating those subjects. So, with that, I'll pass it back.
Thanks, John. Okay, so finally, what we'll do is go through the items, I guess the minimum set of items that you would need to define a target trial protocol, actually in the context of the CVD project. And essentially, so I'll discuss eligibility criteria, treatment strategies, causal contrasts of interest, confounders for assignment to treatment strategies. As I said, that's the minimum information on confounders that you need, the outcomes and the follow-up. And I just want to make 2 points. So I'll go through all of these items for this project. Just make 2 points that I'm not going into detail on any of these. There's a lot more detail in the white paper if you want to look at that. And the second thing is that these are tentative. We haven't actually started working with the data yet. That will start hopefully this week. But these are based on sort of a feasibility assessment that we did the National Biosciences on the kinds of information we can get in their data set in relation to this project. So we'll start with the eligibility criteria. Obviously, we care about the type 2 diagnosis in the past, age, anti-hyperglycemic monotherapy with metformin for greater than 3 months in 2014 or later, which is when most of these drugs the second-line drugs that I talked about were approved for use, or at least when they were all, that was the time when they had all been approved for use. And suboptimal glycemic control after metformin initiation, as defined either by this cutoff that I have here or by a more personalized, individualized glycemic thresholds for each person based on other characteristics that are defined by things like the American Diabetes Association. And as you can see from the source of these, these are all available in the National Biosciences data set from different kinds of sources. We have ICD codes, we have ICD medications or labs, self-reported drug use, prescription records, and so on. For an important point, which is what I've highlighted in red here, is something that we care about as an eligibility criterion, which is no suspected type 1 diabetes or other reasons for hyperglycemia, so things like a rare disorder or pancreatic surgery in the past. And these may be available, but we would have to look at the data again, work with the data set and the database and the people at Nashville to see whether we can identify these for the individuals, for all the individuals reliably in some sense. Because it depends on their start of care at VUMC, and their past history would not be represented, for example. Okay, so the treatment strategies are initiation. So we're looking at initiation of second-line therapy within 12 months of persistent hyperglycemia as measured with HbA1c after metformin initiation from one of these 4 treatment arms or treatments, I guess classes of drugs. GLP-1 receptor antagonists, SGLT2 inhibitors, which as I mentioned is not a part of the GRAY trial, DPP-4 and sulfonylureas. We will also look into insulin if we can, because ideally we want to be able to compare that effect to the GRAY trial because they have glargine as a group that they're using. And other strategies depend on data. So right now we're interested in initiation of second-line therapy. We can also look at sustained treatment with these classes of drugs, and also addition of a third line of therapy, which is often the case in these patients, but they have to progress to using a third line therapy. And this is actually defined in the GRAY trial as well. So the nice thing is we have a reference in the GRAY trial to be able to address and identify these treatment strategies, but again, we're constrained by the data that we have, and we still have to look at the data to be able to identify whether we can have enough sample sizes to be able to estimate these well. And also other confounders for if you're looking at sustained treatment with, and I'll describe that in a bit. And obviously we're limited to drugs prescribed at, dispensed at the VMC pharmacy, which may be different from what the GRAY trial is using. Okay, so importantly, something that we have to define perhaps at the beginning is what are the kinds of causal contrasts, the effects that we're looking into estimating, because they determine the kinds of variables we need on confounders. So we will be starting with the intention to treat effect, which is the effect of being assigned to a treatment strategy at baseline, and this is the effect that's of interest in the GRAY trial, so they're looking at the ITT effect. To be able to estimate this effect, the ITT effect from the observational analog of this, we require adjustment at the minimum for non-random allocation at baseline. So I'll talk about that in the next slide, but also non-random loss to follow-up. So if the loss to follow-up differs between the different treatment arms, we have to adjust for that, because I'm guessing that we don't have complete data on all these patients in terms of their follow-up. The second thing we can estimate is the protocol effect, which is the effect of adhering to the assigned treatment strategy, which requires, in addition to the adjustments that we need for non-random allocation at baseline and non-random loss to follow-up, we also need adherence for non-adherence or non-conformance, so pre and post baseline factors that predict treatment adherence. So we need to know, for example, things like treatment discontinuation and also adverse events and toxicities from various drugs. And this may be feasible, again, depending on the availability of sufficient and relevant data on adherence in the National Biosciences data set. So what I have here are some of the confounders for treatment assignment baseline, basically predictors for treatment assignment that we need to adjust for in our analyses at the end. So things like type of medical insurance, which is available in the National Biosciences data set, history of cardiovascular disease, HbA1c levels, contraindications, and BMI, which are perhaps easily defined variables that are likely to be a part of many real-world data sets. We also have variables like physician comfort level and perception of drug, which may be hard to define. It's probably not present readily, but we will need maybe the help of the treating physician to understand what their preference was, of what sort of criteria they use for prescribing a particular drug, or look at their prescription histories to be able to get to this. And also things like frailty or tolerability. Again, it depends on how we define this, and you may have to involve practitioners. Because this is an important point, I think most people are familiar with the fact that you adjust for confounding if you're using real-world data. Maybe I'll ask Miguel to talk a little bit about if it is ever possible to know whether we have identified all the correct confounders, if you've adjusted sufficiently, adjusted appropriately, and if there are any ways of identifying if you have made a mistake.
And the short answer is no, of course. We have that's always going to be a limitation of observational data. We may do a fantastic job at talking with experts and trying to find out the most important confounders, but we have no certainty that we have succeeded. That's something that we have to live with. That is why we prefer randomized trials, of course. But for the cases where we don't have a randomized trial, or we don't have it yet, and decisions have to be informed, the use of observational data is the best that we can do. Now, having said that, there are some indirect ways of assessing whether we may be very far from a situation in which we have measured and adjusted for all the compounds. One of those ways is using outcome controls. So we can conduct a separate analysis in which the outcome of interest is replaced by a different outcome for which we know approximately the magnitude of the effect, and for which we suspect that the compounding structure is similar as for the outcome that we are looking at. That is our outcome of interest. So if there were some outcome that we know should not be affected by treatment at all, that the effect should be null, and we cannot find a null effect in our observational data, that means we have compounding. So if we have compounding for that control outcome, then we probably have compounding also for our outcome of interest. So a clever use of control outcomes, both positive and negative control outcomes, can support our statement that we think we have adjusted for all the compounders. It's not a mathematical proof that is it may be helpful. There are other things that we can do. For example, if we believe that the people who receive treatment A are very different from people who receive treatment B, and that's so there is sort of compounding, and we may not be adjusting for it well. We can look at the survival curves. See, most treatments have a period during which we cannot see the effect. If we start giving statins to people, we won't see a reduction in coronary heart disease until 6 months to a year after baseline. But if we see in our observational data that the cumulative incidence curves starts to separate very, very early, before the time where an effect is expected, then that would be an indication that there is some residual compounding. So this type of sensitivity analysis can give us an idea of whether we are very far from the mark. But of course, the fact that we can do this analysis and not finding any evidence of confounding, it doesn't prove that we don't have confounding. So the short answer is no. We can never be sure that we have measured all the confounders, but we can do a lot of things to improve our chances of not having a lot of bias due to confounding. Thank you, Alan.
Thanks. Okay, so just for the sake of time, I'm going to skip over this. So outcomes, basically, we're looking at kind of the same outcomes. We're particularly interested in cardiovascular outcomes. This, in terms of follow-up, we have to define time zero, which will be the time of frequent prescription. The patients are followed from time zero until diagnosis of the outcome or loss to follow-up due to various factors. So as I talked about in the beginning, we might have to go back. So if there's a lot of loss to follow-up, we might have to go back and we'll include an additional eligibility criterion of including only patients that have had a history of regular visits as sort of an assurance that they'll continue to do that in the future so that we can get, hopefully, more complete follow-up data for all the patients. So in terms of next steps, we're looking at cohort extraction, sample size estimation, and really all the results. So in terms of the results, from trial emulation, we'll be looking at survival curves and relative risk estimates, and also subgroup analyses for different age groups and different and groups with different CVD risks, for example. And the sensitivity analyses that Miguel talked about, obviously pending we have data against it, and hopefully benchmark our results against the grade. It gets published in time, or if we take too long to get the analysis done. So just in conclusion, in terms of what I've talked about today, in the absence of a randomized clinical trial, try and emulate a hypothetical randomized trial using RealWorld data that can also complement existing trials at a fraction of the time and cost. And we talked about trial protocol design and some best practices for RealWorld data, for how to identify suitable data. And we also talked about best practices for how to design protocols for the kinds of expertise you need for designing the protocol and also working with the big data. And the next webinar, just as a reminder, will take place sometime in August. We'll keep you informed. So we're now going to begin answering the questions submitted during today's presentations. As a reminder, you can still submit the questions. If you go to the questions pane in your attendee control panel, and let's start with the first question. So the first question is, in practice, can we really get an answer for a causal question even if we have randomized study, because it is difficult to conclude a treatment has a causal effect on outcome?
Well, I'm not sure if the question is asking about the philosophy of causation, which I'm not going to get into. But the basic idea of a randomized trial is that you assign people randomly to a treatment, and then you find differences between the two groups. Then the most likely explanation is that the treatment made a difference. That's essentially it. Of course, there are maybe problems if there are losses to follow up, if people are not taking the treatment, or if people are taking the treatment plus something else, etc. So those are always issues. But in science, the best way that we have to make causal inferences is by randomization. When using observational data, we have all the elements except for the randomization, and that's what we need to try to emulate as well as possible. But of course, it makes it more questionable when we don't have randomization than when we do. But I'm happy to talk more about this if the person who asked the question can explain a little bit more what the purpose of the question was and whether I'm missing something.
Mm-hmm. Okay, so we'll go to the next question. Let's take time. Can Miguel please comment on the bias that results in the old way of doing analyses by parsing out person time as exposed versus unexposed?
Well, one possible bias. There are different problems there. One is one typical bias is the selection bias that happens because of the use of prevalent users. There are also forms of immortal time bias that happen there. So let me just give you one example that Alan was commenting on before. If we have data on postmenopausal women that had been randomly assigned to either homotherapy or no homotherapy. We have data from a randomized trial. Okay, if that randomized trial data, we analyze it using exposed person time versus unexposed person time, we are going to get bias. Even though it is a randomized trial, we'll get a lot of bias. Why? Because exposed women that have been exposed for some time are women who have not developed the disease despite being exposed. Those women are less likely to develop the disease in the future. So if we were going to take, say, the first 2 years, 3 years of data of a randomized trial, we delete those 3 years of data. The first 3 years of follow-up, the time from the starting of hormone therapy until 3 years later. If we do that in a randomized trial, and now we compare exposed versus unexposed person time, we'll get exactly the same bias that the observational studies had. Because the problem is that we are losing the connection with the beginning of the intervention, which is time zero. For person time, there is no time zero. Time zero is only for persons. So every time that we take data and we compare exposed person time to unexposed person time, we are at risk of missing everything that happened from the beginning of the intervention until that time where we are looking at. And if there are early effects of treatment, we will miss them. But also we will introduce bias due to the selection of to the depletion of susceptibles, which is exactly what happened in the hormone therapy example. So that is one type of bias. Another type of bias is if we take data from a randomized trial and we compare people who have been using statins for 5 years versus people who have been using statins for less than 5 years or for 1 year, and we classify all the person time as exposed and unexposed using that, we will get bias, of course, because people who have been using statins for 5 years have survived for at least 5 years, right? So that's a form of immortal time bias. The point is that a lot of problems that observational studies have, a lot of observational studies are published and are wrong. And we can prove that they are wrong. And they are wrong even though they are published in the best medical journals. But they are not wrong because they are observational. They are wrong because we are doing the wrong analysis of the data. If we were doing that analysis with randomized trial data, we would also get wrong results. And that's a very important difference. That when we talk about the problems of observational data, if you look carefully, in a large proportion of those observational studies that gave wrong results, the problem was not lack of randomization, was not confounding. The problem was that the analysis was almost guaranteed to give you wrong results, whether you do it in an observational study or in a randomized trial. Back to you,
so
Anna.
So you have a lot of questions. Just like one final point on missing data. Are there concerns with imputation? What do you do with missing data?
Well, it depends where the missing data happens. Of course, if we have missing data for the treatment of interest, then we definitely don't want to impute that. We will have to restrict our analysis to people who, at baseline, have data on the treatments of interest. If we have missing data on the confounders, then that is an interesting one, because see, our main concern is that we want to be able to emulate randomization. So we need very good data on the confounders. We have lots of missing data on the confounders. We are probably going to fail. And also it doesn't make a lot of sense to impute the data on the confounders, because the data on the confounders, what we impute will be using data from the other confounders. But that's not going to help us adjust for confounding, because at the end of the day, we're just using data from the confounders without missing data to impute the data on the confounders with missing data. So in most of our applications, what we do is we restrict all the analysis to those without missing data for the treatment and for the confounders. If there are some confounders that are not very important, or we think are not very important, and they have low levels of missing data, okay, we may create some missing data category for those people and include them. But if we are looking at the effect of statins on heart disease, and we have missing data on statin treatment, those people cannot possibly be part of the target trial emulation. If we have lots of people with missing data on LDL, well, those people cannot possibly be part of the target trial emulation because that is the key confounder. So missing data, if there is lots of missing data, we will have a smaller effective sample size for our emulation.
Thank you, panelists, and thank you everyone for attending today's webinar. And on behalf of Cytel, thank you for joining us today, and have a great rest of your day. Bye.